So this next top is about the laboratory exercise that most of you are going to perform tomorrow. And again, we're going to sort of do almost all of the steps there as shown here. The assay barcode import, I'll just show you a demonstration, but you guys will actively do all of these steps here. So that'll give you practice using the registration wizard. I'm not going to torture you with manual setup. I'm going to let you guys just do it with the Janus. And then we'll get practice setting up and running the IBIS T5000. So we're going to make 2 types of plates. We're going to make a total of 6 plates in all. So we'll partner you up and you'll each make part of 1 plate. You'll either be making a blinded sample plate, so you won't know what your sample is, and your goal is to correctly identify the sample by searching the general database and see if you can match it. The second one would be a known sample plate. You know what your sample is, and now what you want to see is, are you getting a reproducible profile? We've already registered the profile. You have CS30. When you compare it to CS30 that's already registered, did you get the same profile? So this might be sort of like comparing an evidentiary sample and a known sample, but in this case they're the same sample. So this is the import wizard example. You select this from IBIS track, and it allows you to import your assay kit barcodes or your assay kit configuration, and it automatically knows what you're trying to do by the type of file that you select for import. You then just hit next button after selecting your file. If you're importing barcodes, it'll show you all the barcodes that are being imported from that file and their expiration date. The system will still let you use them, but it will flag them once they expire. And you click import, you'll get an importing has completed, and then you'll click finish. And so you've imported your resources that you need to now register your experiments and do your experiments. As we've sort of emphasized throughout the day, when you register an assay, you register 10 samples and a positive and negative PCR control. The data analysis actually looks for the positive control and negative control and makes some judgments about the quality of the plate based on these. And following slides will show an example with the Casework wizard, which you'll get hands-on experience with tomorrow, setting up 10 samples from tubes. So the first thing you do once you select the Casework wizard is you select your assay kit. When you select your assay kit, your inventory should show that you have barcodes available. If you have 0 barcodes, you need to go back to that import step. You won't be able to register any plates. You then can select a project and select or type in an experiment and hit the next button. You can run a variety of different sample types. You can run unquantified, quantified mass if you've done quantifiler, or quantified copies if you've done some of the mitocopies assays out there. When you're selecting your plates, if you select unquantified, there'll be a dilution factor entered for your PCR plate. If you select quantified, either picograms or copies will be entered as the unit on the PCR plate. Again, you use an Excel file, and here I have an example of 9 hair samples and the reagent blank that was done while the samples were isolated. And in the quantity, I have 25. Since I selected unquantitated, this means I'm diluting these samples by a factor of 25. And so your samples will show up once they've been validated. Then you can select a control layout. And what that does is it interleaves the negative control and the positive control based on what you've previously arranged in your control layout. And this is something that the user can determine and select. Then the next step is you select your PCR barcodes that go with it. The wizard tells you how many you need to select. You just highlight and click the left arrow and assign the barcodes. The next step, at the completion of this wizard, you'll have several files that will be generated, and you need to select the location where these files are going to be stored. You'll get a report file and then some work list options. If you're working with a Janus, you want to select this option so that you'll register with a setup plate, because the Janus requires that you have a setup plate, so it has a barcode associated with where your samples are. As an intermediate step in the registration, it'll tell you the PCR barcode for your assay plate and then your S barcode for your setup plate. And it'll show you this report here that shows you your sample ID, its dilution factor on the plate. If it's from a tube, it generates a location for you that's arbitrary. But what you're concerned about is the setup plate, which is where it goes on the Janus to be set up correctly and put into the correct column on the PCR plate. You'll have to go and find these reports. You want to print out the report file here, because that'll tell you the barcodes that you need to set up your Janus. If you don't have an intranet, then you will need to copy this Janus file onto a jump drive so that you can copy it to the computer where your Janus is, because this file will provide the information that tells the Janus how to do your sample layout. And this file here just gives you some instructions on how to set up, where to put samples and plate locations on the Janus. So one of our concerns in our lab is obviously contamination control, and as most of you probably do anyway, we control that by gowning procedures and decontamination of our work area. So I will have you guys gown up tomorrow in a lovely blue bouffant, disposable lab coat, booties, face masks, and gloves, and I even have safety glasses for you guys. Prior to you guys coming in, the people that work for me will decontaminate the PCR hoods, pipettes, and consumables. We do this in a two-step process. First, we spray the work surface with bleach and let it set for 5 to 10 minutes before wiping down. Then we also wipe down our work surfaces with DNA erase, and we wipe down all of our pipettes with DNA erase as well. Then we'll place any new consumables we want to use, such as tips, tubes, or reservoirs, and turn on the UV light for 15 minutes and treat everything that's in that hood for 15 minutes. We also actually use a sterile wipe that's doesn't tear. It's a nylon base, so it's not as absorbent as a paper towel, but it has the advantage that if there's sharp edges and stuff, it won't shred and leave paper behind. And that's nice because we do these first two steps also on the Janus. We can't UV treat the Janus because it has tubing, and the UV light would degrade the tubing and cause problems. So then we're going to have you guys actually set up PCR plates, when the first step is actually sample dilution, because you do need a minimum of 50 microliters for each sample to set up your plate. So 40 microliters is required for the actual sample, and then we have 10 microliters for waste, and a new tip will be used each time you're adding sample to the well. We'll perform the sample dilution in a PCR hood. For quantitated samples, we recommend that you go down to 500 picograms per 5 microliters or 100 picograms per microliter, because that gives a very solid profile without using too much sample. For non-quantitated, you just need to do whatever dilution you feel comfortable. We're currently doing a 1 to 3 dilution on some of our samples, and on tomorrow's samples, we'll actually be doing a 1 to 10 dilution on them. Then we'll set up the Janus and have that do it. After the Janus is set it up, we're going to put this Super Pierce heat seal. We'll vortex the plates to make sure your template mixes in your PCR master mix. We want to centrifuge it to make sure that everything's down at the bottom. And then now the plates are ready to be thermocycled. We recommend using Eppendorf thermocyclers. They're what's validated with our assay. The ramping speeds and times are set to work with our assay and maintain our balance. Also, our plates fit in them. Our plates won't fit in some of the other thermocyclers. The thermocycle program is in the neighborhood of 3 hours and 16 minutes. We do 36 cycles. We also have an additional 10-minute step at the end at 99 to heat kill the enzyme so that you don't get enzyme activity while it's sitting on the T5000 and getting that non-templated A. Because we've had cases where, you load the instrument up for the night and unfortunately something happens, you do it wrong and you knock your hotel over and your plates will sit for 24 hours before you come in and reset them. And you don't want that non-templated A occurring while they're on the deck. You can freeze the plates before putting on the IBIS T5000 if you're ever in a situation where you're thermocycling faster than you're running on your T5000. And as we discussed a little bit on the tour, you can probably do that for about a month, freeze your PCR plates before running them. We'll then have you guys load the T5000 from the tour. You know that there's 3 different hotels, one where you put your crude PCR, your magnetic bead, and your elution plates. The one key thing is that the barcodes must be facing the arm, because the arm will go and read the barcode and ensure that your PCR plate is registered and that your mag bead and elution plates are kit, which means they haven't been used, because you cannot reuse these, the mag bead or the elution plate. Those are truly consumable. They're used once and discarded. We'll then fill the reagents and empty the waste, and we'll start the T5000 controller software. Since we'll have just added reagents, we recommend priming the system, which is just selecting this button and following the directions on the prime. That ensures that there's no water or no bubbles in your system water or in your reagents that could cause problems downstream. Once you hit the start button, you'll be asked to enter a number of plates to be run. You can put anywhere from 1 to 15. The next question will be, do you want to allow a system QC to be formed? If you're running your instrument every day and starting every day, you don't need to do this every day, but if you've gone over the weekend and haven't run it, we recommend you doing it at the beginning. This ensures that you're actually still getting mass spec signal and that it's being acquired and that your CR3 reagent is looking good. So what's going to happen tomorrow is we'll start out here and we'll actually register plates as a group. Then we're going to divide you into 2 groups. One of you will come with me back to the labs and start the lab exercise. The others of you will remain behind and get data analysis practice on computers with Tom. Then we'll switch in the afternoon so that both groups get to do both activities. And then in the afternoon, after group 2 is done, we'll actually set up the T5000 as a group. And then on Wednesday, you'll analyze the plates that you guys actually made using the software again. So we'll register the samples here, and then we'll do the PCR setup with the Janus. And like I said, we'll already have the hoods and supplies ready for you to go.